So we need to think about that in terms of our compounds. If we focus purely on microsomal stability, this can lead to unexpected metabolism by other enzymes in late-stage studies. To help us to think about that, we have also developed what we call the which enzyme model. This identifies the enzyme families that are most likely to metabolize a compound, by analogy with the which P450. So again, represented by these pie charts here, you can see the relative likelihood of metabolism of a compound by each enzyme family. And this can really help to prioritize appropriate in vitro and in vivo PK studies. A couple of actual examples of this are two clinical candidates, felnitamol and cabazitaran. These were actually candidates that failed in the clinic due to poor oral bioavailability and exposure, despite the fact that they had passed all of the usual preclinical PK studies. So let's just look at the prediction of metabolism by these compounds. I'm just going to jump back into the software. I'm just going to bring up these two molecules here. And again, I'm going to predict the metabolism of these compounds. Again, the results appear instantly just for convenience because I have pre-calculated those. And what you can see here in terms of the which enzyme and which P450, you can actually see summarized in the table as well that these are predicted to be largely metabolized by aldehyde oxidase, not by P450s at all. So here you can see felnitamol, here you can see cabazitaran, again, predominantly aldehyde oxidase. And indeed, we can also predict the sites of metabolism. So here you can see for felnitamol, there are actually 2 sites of metabolism that are predicted for aldehyde oxidase. And we can even drill down a bit further and look at the details, and you can see the most likely site here, the statistically most likely site is this position here, which has a slightly higher probability than this position. So we can, again, provide that guidance as to where the compound can be modified to improve that metabolic instability. So in this case, these predictions that aldehyde oxidase is a very likely candidate for metabolism of these compounds would lead to choose some different experiments to do earlier on in drug discovery, not wait till the clinic, but for example, looking at PK studies in guinea pig or rhesus monkeys. They have good aldehyde oxidase activity, whereas rats and dogs have little or no aldehyde oxidase activity. So this would help to prioritize those in vivo experiments, or indeed look in vitro at hepatocytes, for example.